All right, so let's start with thinking about what we worked on yesterday. So yesterday, you guys were having a chance just to explore genetic diversity as a big picture concept, and I had you guys read an article to find out basically, range of genetic diversity. What does that then mean? What does it mean to survival and things like that. All right, so my jar is coming back out because I like to pick people randomly. If you don't know, that's fine to say, I don't know, I don't remember. But I just want you to have a chance to share one thing that you found interesting from the article you read yesterday. So person sharing is Julie. What was one interesting thing about that article? Yeah, Liberian lynx has the least genetic diversity. It was something like 0.028%. So what that tells me is that these lynxes are very genetically similar to each other. And I shared this fun fact with some people, but I'll share it with the class because I think it's interesting. You know how in humans, if you were having an organ transplant, so let's say someone was sick and they needed a new liver, I can give them my liver, or if they need a new kidney, I can give them part of my kidney. The person that takes my donation needs to take a drug that's known as an immunosuppressant. So it suppresses their immune system. And the reason why we need to suppress their immune system, most people think that's a bad thing. I want to have my immune system to work so that way I don't get sick and I don't get COVID. In the event of having a new kidney that's from someone else, we need to suppress the immune system because we are genetically different enough that if someone has my kidney, the body will recognize it as foreign and kill it and reject it. So it rejects the organ. You sometimes hear about that in organ transplants, that rejection of the organ, which is a big concern. They do their best to try to match people as much as possible to limit the risk of you rejecting that organ, but it does happen at times. However, in species that are not as genetically diverse as we are, say for example, cheetahs, you can transplant an organ from one cheetah to another cheetah, and you don't need to give them any immunosuppressants. They will not recognize that kidney as being a foreign object. They will think that that kidney is themselves. So that kind of just shows how little genetic diversity there is in the cheetahs if they can transplant organs without any immune response to it. Yeah? Which would be really clinically bad. We saw that with our our Buzzy McDonald example, that if there was very little genetic diversity, all of them would die if there was one stress point. Cool. All right. Anybody else want to share? I'm gonna pick someone else out of the jar, but I don't want you guys don't feel like you're speaking on it. Yeah, what's up? What's something that you learned that was interesting? That mushrooms, I'm going to remember what percentage it was? Isn't it cute? So they're so very different from each other. So in that case, it would be a problem if we were doing any transplants with mushrooms, but we're not because we just eat them their own. All right. So for today's session, we're just wrapping up the lesson that we had yesterday. I'm totally fine that we spent the whole period looking at that because it gave you some big picture ideas. But now let's start diving into the why. So we need to understand what is causing this genetic diversity. So we need to think about protein and we need to think about DNA for that. So lesson plan for today, we scratched up the first two. So what we're going to be doing is the remaining things. I'm not really sure how this is going to time and with the lesson and with the activities that I planned. So we're definitely going to get some notes on the relationship between phenotype and protein. I'll give you guys some side tab pages to reinforce those. Some of you guys have already probably done them, so I can find you something else to do. We'll also talk about protein to DNA. So there's lots of different layers to how this works. I'll show you a video on the discovery of DNA because I think it's good to know the history. And we also have a Kahoot at the end for the DNA structure, which I thought would just be some fun. Number 9 then is I'm not really sure what direction I want to go in yet. It kind of depends on how much time we have. I really don't want to rush these two activities on genes and then another bracelet. So we might move that into my next lesson. This actually works probably better with the next lesson. So we'll see how we go. If we do have some extra time, I'll start showing you a documentary on the secret of photosynthesis. which ties into the discovery of DNA, because there was a lot of controversy when that DNA structure was discovered. And I think it's good to know the history, and it's also good to be learning about a female scientist. All right, so for success criteria, 2 things. So on that orange piece of paper, we're welcome to tick off covered in class that we worked on number 1 and we worked on number 3. We're looking at, like I said, defining some of those key terms like heredity, genetics, and traits, and we're also exploring how that genetic diversity is beneficial to survival. Cool. All right. Phenotypes and proteins. So these notes here are in the booklet. So I don't want you to worry about scribbling down everything that's on the slide because it has been provided for you. In fact, I suggest you get the booklet open if you're planning to do paper notes and write onto the booklet itself. This is for university, which is very similar to what I did at university, and I'm sure it's probably similar to what you guys will do as well. Lecturers provide the slides, and you can jot down things as you're listening. Unless the lecture is really mean and they don't give you any slides, and then you're jotting down things like a crazy mess. If you are doing your notes online, you feel free to open up the PowerPoint alongside with your notes document, and you can copy and paste any images you want to cross or the slides. You don't really need to because you do have a paper copy. Does anybody not have a booklet because they joined the class later or were absent and handed these out that need one? No? We're good? Okay. Let's start talking about phenotypes and the relationship with the proteins. We know that the phenotypes are the physical traits that are presented, such as, the brown hair. But the phenotypes that we have are the result of something known as a protein. Proteins have a huge wide range of what they do and their functions and their shape. You guys have heard of protein before, like you're like, oh, protein shakes. Oh, I need to eat some protein. I need to eat some meat so I get protein. We have to think about those sort of things. But we're going to think about them in a little bit more detail and a little bit more structure. Proteins have a whole list of different actions, and I've listed them out. Okay. So these are a list of different functions, and I'll go through some examples of each one so you know what they mean in context. So these are just examples, and we can see by their function why it then has that phenotype. The first one is enzymes. You may have heard it last year, you probably should have, with catalysts and things like that, but this is chemistry related. Enzymes are catalysts. Catalyst is just a special word to say speeds up chemical reactions. You don't need to be a protein to be a catalyst, but if you're an enzyme, you're definitely a protein, and you're definitely a catalyst. What these guys do is they have a very specific shape, and with that specific shape, items are able to fit inside, and it can then help that chemical reaction occur. So they speed up chemical reactions for us. Enzymes are a really good example. You have a lot of them in your gut to help break down foods, that way you can absorb it. They're also used to break down sort of drugs and things like that. And we can actually inhibit enzymes to prevent the breakdown of drugs. So that's the first example. And feel free to stop me if you have any questions, if you want more clarification. Here's an example of enzymes, and I love this example because it relates to me, and that is the lactase enzyme. Is anybody here lactose intolerant? I'm one of them. So you can't drink milk without getting really bloated and like sick. No? You guys are lucky. It's up. I can't eat cheese. As you get older, you start losing that enzyme because you're not drinking the mother's milk anymore. And so if your body can't break down the lactase sugar, the bacteria start doing that. And when the bacteria do it, they produce gas and irritation, and that's why it's not a fun time. And that's all because we are missing this enzyme here, which is known as, lactose is the sugar, lactase is the enzyme. So the enzyme lactase breaks down that sugar. Lactose intolerance is quite common in Asians, about 90%. So 90% of Asians don't have the enzyme to break down the sugar. So, and that reflects our diet as well, because we don't have a lot of dairy in the diet. So we'll see when we're looking at the larger population genetics how things like the lactase enzyme evolved in certain populations and not in other populations. But that's an example of a phenotype. The phenotype is I can't break down the lactose sugar, and so I will get this. The good news is you can actually buy pills with the lactase enzyme. You can take that while you're eating the milk, and you're fine. So that's the first example. The second example is a transport protein. So what these things are responsible for is moving things around the body. The example that I have for you is the hemoglobin protein. Hemoglobin is found in our blood cells, and what they do is they transfer oxygen around our body. It's a very important very important protein that fits, and it provides a space for that oxygen molecule to attach to. Otherwise, it's not going to dissolve into our blood. Structural proteins, these are the ones that you probably can see and think about the most, and that's things like hair, horns, feather, quills, skin tone, all those things. So that's why we have that variation in our hair structure and things like that. It's because of the protein that's being produced. Are we good so far with that? So I want to show you that with these proteins, there are going to be things that you can physically see, and there's going to be things that are happening internally that we can't, but are still very important to function. Hormonal protein. These are basically like little messengers around the body. So our body is huge, and we have all these organs, and we need them to work together, and we need them to do certain things. The way our body tells each other to do things is by releasing hormones. Now, you guys probably know most hormones like estrogen and testosterone. Those are our sex hormones. But there's other hormones such as insulin that regulate blood sugar levels. They tell the body when to release sugar from our storage or to absorb sugar from the blood. If you have type 1 diabetes, you don't produce this hormone, and you have a hard time in regulating your sugar levels. So again, that's, like I said, a phenotype, not being able to regulate your sugar levels. Next one are our contractual proteins. Those are the ones that are found in muscles and allow for movement, and that's why we see us referring to meat as protein. Defensive proteins we are talking about in the context of the immune system, and these help us protect against diseases. These are what we know as antibodies. These, again, with that very specific shape and structure, it's able to recognize foreign objects and then surround them so that way they can't replicate, they can't move, and then it makes a signal for our cells to eat it and kill it. So that is what we're thinking about a lot when we're thinking about our immune system and with COVID and vaccinations. It's all about teaching the body to make these proteins. Are we good so far? Questions? Do we have an idea of what I mean by protein? So the phenotypes that we see are the result of the protein. Cool. So the next thing, as I mentioned, the important thing about proteins is that they have a very specific shape, and that very specific shape allows them for that function, to have that function. So the question now is, why do these proteins have these specific shapes? So we need to then talk about the structure of a protein. So protein, as a starting base, is a chain of amino acids. And what I have on this right-hand side of the PowerPoint is a list of different amino acids. These amino acids have different molecular sizes and different properties. So the amino acids have a specific repeated section, which is not highlighted. So all of those amino acids have that same structure. If you take level 3 chemistry, you'll learn more about what that structure is. What is highlighted in pink is what is unique to that amino acid. So that gives it, we can also see that they're different size molecules as well. That's going to then influence the shape that it can then fold to. So, fun fact, humans need 20 amino acids. We only create 12 in our bodies. So the others need to come from our diet. So we eat and ingest the rest of the amino acids we need. That's why it's really important to be kind of mindful of your diet, so that way you're ingesting the amino acids so you can keep building your protein and things like that. Their shape. The first thing, like I said, is that the amino acids are a chain, the proteins, as the first part, are a chain of amino acids. So it's kind of like a necklace with a lot of beads. So that's what we see in our first structure. Now what we can see in that first structure is that there's some of these beads that are orange and some of them that are blue. So one of the properties that I mentioned earlier was hydrophobic and hydrophilic. Hydrophobic means I am afraid of water, or I do not like water. And some people do have hydrophobia. I'm kind of in that boat. I'm not a huge fan of water. Hydrophilic means that they are water loving. So that's going to influence how that protein folds. If you think about your cells, do you think this inside the cell there's a lot of water? Yes. So if I'm looking at this structure, which color do you think will want to be on the outside near the water? And which color will want to be on the inside away from the water? Orange would like to be next to the water, and blue would like to be hidden away. So we can see that with how the protein folds. So we see this tertiary structure here, that the orange is all on the outside, and these blue bits are hidden on the inside. And that's what's influencing the fold and the shape of the protein, and thus giving it particular structure and function. There's other things to kind of keep in mind. There's lots of repeated structure like beta sheets and alpha sheets, which you can see here is the curly kind of bit and that little foldy bit. And then sometimes multiple tertiary structures of that fold will join together to make our full-blown protein, and thus our function. So that's what we're looking at with why they have those specific shapes. If there is a mistake in the sequence, it could then influence the protein and make the protein not work. So yeah, one change of an amino acid could affect the folding of the whole protein and make the whole protein defective. A good example of that is sickle cell anemia, and we will look at that as an example to what that mutation is and how does it affect the individual. All right, let's take a break from me talking. Some of you guys already have this iPad page. If not, I'll pass it out. And what you are looking at is just some information on protein folding. So if you're going to take a little break from me talking, if you finish the sheet, I'm happy to give you a sticker. If you have already done it, because you started yesterday, I want you to start researching chirons. They're neat and also kind of scary. And you can write a little paragraph summarizing what you researched. So that's what I want you to do if you've already finished this iPad page. Cool. Feel free to listen to music right now while you're working, and then I'll move us on. Let's go. It's a door. All right. So, there's lots of new vocabulary that have to be mindful of when it comes to proteins and biology in general. So, now that we know the importance of protein, we can now start looking at, okay, how do the proteins know what amino acids to grab? So, we're back and we're going more and more detailed into our thought process. So basically, our next question is, how do we know what is the sequence of the amino acids? So if you guys remember what I was talking about, that sequence of amino acids is the first step. And with those sequence of amino acids, it really affects how that protein will fold later. So, sequence of amino acid is determined by the DNA. So the DNA holds the code of life, and it explains these are the amino acids to grab, and then the cell then folds them. So we refer to DNA often as the code of life. It's like a computer code, but the computer code for life. What I have here in an image, and we'll talk about this in another lesson, is basically we have a cell. Inside the cell, one of the organelles is the nucleus. So organelles are basically kind of like the organs of the cell, thus organelle. When we do microscope stuff, we'll talk more about the organelles. Inside that organelle is the storage of the DNA. And the DNA is this double helix strand that we are looking at here. Now, this has been folded into a chromosome. So basically, you have a long string, and they kind of rolled it up into a ball of yarn. So that way, that's our chromosome. Don't worry about those vocabulary too much because we will go through it in more detail. And more specifically, I just want to give you guys some reference point to what you're looking at in that image. Cool. So, like I said, the sequence of amino acids we grab all depend on the DNA and what the DNA tells the body to grab when it comes to those amino acids. So in order for us to understand that, we need to refresh the DNA structure to understand, okay, how is it actually acting like a code? So let me grab my document camera. You guys would have seen this image from last year because you normally fill this in as like an achieve level question on the assessments. So let me kind of walk through that slowly with the different steps. I did print out a copy for myself to write on. Cool. Camera and get my markers. Once I find my markers. Cool. All right, so here's the structure of DNA. The first thing I kind of want to note is the different components that we are looking at. There are three main components, and I'll talk about how they all kind of fit together. The first one that we're looking at is that P, and that P is referring to a phosphate. The phosphate is one of the components of our DNA. The other component of our DNA is these little houses kind of thing, these pentagon shapes. I'll do it in a different color, and I'll kind of circle it so that way you know which one's which. Second thing then is our sugar component. If we want the fancy name for that sugar is deoxyribose is the fancy name for that specific type of sugar. Next. The last thing when it comes to our DNA component is the bases. And so that is this section here. Now with the bases, you would have heard them as 4 different types. There's adenine. Let's see if I can spell it. Actually, go to my PowerPoint. I do have them spelled. All right. So there's adenine, which we just abbreviate as the A. There is also thymine, which I don't think I spelled right. I wrote thymine, which we abbreviate as a T. There is cytosine, which we abbreviate as the C. And then the last one is guanine, which we abbreviate as the G. So those are my different bases. And what you should hopefully notice is that the bases have a very particular matching. So A always matches to T, and there's 2 hydrogen bonds there that hold that together. Don't need to know that. I'm just adding that in case anyone's doing chemistry and we talk about hydrogen bonds later. So it could either be in either side. A is on that side, T is on that side, and this side T is first and then A first, but those 2 always match together. And then guanine and cytosine are the ones that match together for the other pair. And they have 3 hydrogen bonds. Are you okay with that so far? Yes or no? Does this ring a bell from last year, hopefully? Cool. All right. So that's how they pair, and we can see there's some guanine and cytosine down there, as well as some examples. So that's really important because that's giving us the code. The other important word is nucleotide. So when I say the word nucleotide, I am talking about all 3 components together. So a phosphate, a sugar, and a base. Don't worry, I will scan these notes and put them online. Nucleotide. So nucleotide is specifically all 3 of them. So you'll often hear things being referred to as nucleotides. This here is known as our phosphate backbone. And we can see that on both sides. The other thing I want to point out to you guys is that these guys are anti-parallel. They're going in two opposite directions. You notice in this case, the oxygen's on the top and going that way, and in this case, the oxygen's on the bottom going the other way. If you decide to pick up any of the extra standards, you'll need to be able to talk about that. Are we good so far? But in this standard, you don't need to know that. You just need to know the basis. Questions? Moving on? Okay. Don't worry, I will scan it and put it online. Oh, wait. Yeah, you can have it. It's back, but you can have it now. All right. So, I thought it'd be cool to have some fun facts about DNA. About DNA is that once my clicker's not going to work until the clicker gets plugged back in. Very basic understanding. Okay. Cool. All cells in our body have DNA with the exception of red blood cells. If I took all the DNA and all my cells and I unwrapped it, so that way it wasn't coiled together and it was all like one long string, it would reach the moon back to Earth 600 times. That's inside our body. Obviously, it's really, really tiny. Are 99.9% identical. We do obviously have some variation, that's why we don't all look the same, but we are obviously quite similar as a species. Our genome, if we were to translate that into data, it would be 3 gigabytes of data is stored in our DNA. And the other thing that's interesting is about bringing back extinct animals. This is often talked about as a possibility, but that is only possible if the DNA is less than 10,000 years old. If it's older than that, it's too degraded, the quality is too low, and we can't bring those animals back. So I'm sorry to break this to you, but Jurassic Park is not possible. You can't get the dinosaurs back, the dinosaurs are too old. But there has been talk about bringing back other extinct species such as mammoths and things like that because they fit within that range of 10,000 years. Since there's no current mammoths to impregnate, they probably would impregnate it into a similar animal like an elephant if they were to bring back. So, some fun facts. All right. Do you guys want to watch a video first and then do some more side pack work? I also have the Kahoot as well, so I don't really know what more you guys want to do things in. Do you want to do the Kahoot first? Yeah, okay. We'll do the Kahoot first, and then we can backtrack and do other things. I don't know, I have to look it up. I can tell you in a moment. All right. So we have roughly 10 minutes left. So what I will do is I really think it's important to watch this video to know the history of the DNA discovery. But I also want to give you guys the option to get some work done. So what I'll do is I'll pass out this DNA worksheet. You're welcome to work on it while you're watching the video. I'll still give it another sticker for the completion of the DNA worksheet. If you guys remember my policy and homework, some people have been asking me on it. I don't assign any homework. Basically, what homework is whatever you finish in class or whatever you want to do extra to extend yourself. So stuff like this worksheet would be stuff you could do for homework. I'm happy to give stickers for it later. So if you see me next period and go, oh, miss, I finished this at home, I'll give you a sticker for it being done. Okay, so it doesn't need to get done in this class. If you are ever looking for more ideas of what to do, go to biology. And we're going to go to the second lesson here. If you guys are ever looking for more work, you've had it enough. Look here where my Mahi choice is. And I have things at different levels. So you can level up and you can start at any level depending on your confidence. So most things you guys actually have done because I've given you the DNA recap and I've given you the protein structure and DNA duration. But if you want to extend yourself, you can do the research on the prions. You can just do more research on the discovery of DNA. You guys actually have done the level 3, which was those questions from that website. So I do give you guys more work on the website if you need anything. But I like to give you guys choice and that freedom. Anyway, so we'll watch this video, but I want to pass out the. Some you guys saw it the other day. Yeah, this is actually the one I gave you because it's supposed to go before, but I wanted to go the other direction. All right, so while you guys are working, I'm going to play this video on the discovery of DNA because I think it's good to know the history of it. Feel free to work on that worksheet while this video is playing, and then I think that'll put us to wrap up. What is it? Tuesday. Am I wrong? I actually have much more time than I thought I had. I'm going to be until 11:10. I'm not going to make that mistake again. All right. So we actually have 15 minutes, which is fine. All right. I'll show this video. You can be able to get that sheet done before the end of the hour. Anyway. All right. So we have about 10 minutes left in class, and if I think you want to keep working on that DNA worksheet and get yourself a sticker for that, but I feel like we just kind of want to chill and zone out. I'll start this documentary on Rosalind Franklin. You probably won't finish it, or we won't pick it back up in another lesson, but if you do want to finish watching it, it is on YouTube. It's called The Secret of Photosynthesis 1, and it goes through Rosalind Franklin's life and like how Watson and Crick basically stole her work and took credit for a lot of the work, and just basically the race of discovering the structure of DNA. So it's really good if you just are curious about the history. So I'll play some of this. I might also kind of wrap up today's lesson before I start playing it, so you guys can kind of pack up if you want. Protein and DNA. If you guys successfully pick the material for this lesson, you should be able to define hereditary genetics and traits, and you should also be able to explain how genetic diversity is beneficial to survival. So I'll put on the documentary so you guys can kind of get a start and see it. You're welcome to keep watching it at home. You're also welcome to keep working on that